So before we're actually going to talk about the electron transport chain in details, I would like to take a minute and to explain you guys the actual purpose and the reason why we need the electron transport chain. So the electron transport chain is basically a sequence of proteins that you can find on the surface of the inner membrane of the mitochondria. So this is the matrix. This is the intermembrane space of the mitochondria. And you can find a sequence of different proteins on the inner membrane. Now, the following proteins are basically acting as an electron acceptors. They are going to accept the electrons from the electron carriers we talked about a lot, from the FEDH2 and NADH. And once they accept the electrons, and they move the electrons in sequence, in chain, from one to the next, you are going to see that the movement of electrons is going to yield energy. Because the movement of electrons is going to yield energy, we are going to be able to actually do something very special. The energy that is basically being generated from the movement of electrons from one to the next proteins is going to cause a conformational change in the actual proteins that we have. And some of the proteins in the chain are going to act as a pump, as an active pump actually. Now, the proteins that act as a pump are going to pump a very specific molecule, a very specific particle outside of the electron transport chain, outside of the matrix. And the particle which is going to pump outside is a proton. And the following first, third, and fourth that we are going to see in the next videos, proteins of the electron transport chain, once they have enough energy from the electrons movement that they were donated, they accepted from the electron transporters, are basically going to pump protons from the matrix to the intermembrane space. And we are going to generate something called electrochemical gradient, because we are moving the protons against the concentration gradient. And what we are going to get is an accumulation of positively charged protons on one side, comparing to the lack and the low concentration of protons in the matrix itself. We call it electrochemical gradient because it's because you have the difference in the electrical charge between the two. And we also have the chemical, which is the fact that you have a specific particle, a specific chemical on one side more concentrated than the other. So we have electrochemical gradient. And because we yield the energy to move and drive the particles from the matrix to the outside, we are going to have a lot of potential energy that we can use for a different thing. And you will see that the accumulation of many particles, many hydrogen, many protons in the intermembrane space is going to be yielded into a very specific process. And this process is the movement of the particles through a specific enzyme, a specific protein that is basically going to help us to generate the enough energy to take ADP and to phosphorylate it again to ATP. So the fact that we are generating a lot of protons, a lot of electrochemical gradient that is going to help us produce a lot of potential energy, which we are eventually going to use in order to drive the phosphorylation of ADP into ATP, is the fact that is the reason that you can drive and to create so many ATP molecules in cellular respiration itself. Because you will see that this process requires oxygen. This is why it is an aerobic process, and it is the most effective way to actually produce ATP inside a cell, because you see the vast majority of ATP that we can actually use out of the 38 ATP we talked about in the introductory video can actually be produced in this process called oxidative phosphorylation, because we are going to take ADP and phosphorylate it again to ATP using oxygen in this sequence. So I'm going to sum this video up very quick. We are going to learn in the next few lessons on something called the electron transport chain. And the electron transport chain is a sequence of protein that you can find on the inner membrane of the mitochondria, which is basically, some of them are going to be able to accept electrons. They are going to accept electrons from the NADH and FADH2 that we are getting from the Krebs cycle. Once they accept electrons from the coenzymes that basically carry the electrons, it gives them energy that they are going to use in order to drive and to pump protons from one side to the other side. This is going to cause something called electrochemical gradient. And because we have this electrochemical gradient, the gradient gives us a lot of potential energy that we can actually yield and to take advantage of. The mitochondria is taking advantage of the fact that it has a lot of electrochemical gradient in the intermembrane space comparing to the matrix by moving the protons through a specific protein which we are going to learn. And these protons, when they move through this protein, they are basically giving energy to this protein to phosphorylate ADP into ATP again. We're going to study everything in detail. I know it's a bit complicated, but in a nutshell, this is basically what you need to know for the IMAT and BMAT exam when it comes to oxidative phosphorylation. Next video, we are going to actually analyze the electron transport chain, and we are going to see what are the names and which structure we have on different sides of the electron transport chain. This electron transport chain is actually something you should know in more details, because when I analyzed the past papers, I saw many questions that were quite specific for the electron transport chain, because it's a very high yield topic to know in the medical field as well. Because, for example, you can block different places inside the electron transport chain using specific drugs and specific toxins, and you will see that you are basically blocking cellular respiration and you can cause death and necrosis, which is cell death, which is very important when it comes to the medical field. So in general, you are expected to know the electron transport chain way in more details than what we actually studied when we talked about the Krebs and the glycolysis. I hope this video made sense. In the second part of this video, I'm going to actually explain the following illustration, where we are going to actually understand which are the electrons and where they are going in the electron transport chain and how they actually yield the energy to drive the protons outside.